into a bar, orders a beer. Bartender says no. Retrovirus replaces the bartender and says, now you do. So this is mostly what I'm going to talk about today, is the viruses, which is in our DNA. And I'm going to show you that what is the reason why we have 8% of our DNA derived from retroviruses. So these retroviruses in our DNA, they are so crucial for early human embryogenesis that when we put the 3 base pair mutation in the regulatory regions of these retroviruses, we saw that these embryonic stem cells, the pluripotent cells, if you will, which is the mother of all the lineage of human body, they just undergo apoptosis. They do not survive. And the reason is that because these retroviruses, they invade us, they become part of our DNA with the enormous regulatory potential that there's not much options for the host than to co-opt them. Because during early human embryogenesis, we are exposed to lots of pathogens, lots of the materials which are ready to kill our embryo. So host has to evolve. Host has to find some additional defense mechanism, how to defend our system against those kind of pathogens or retroviruses. Well, when we talk about protein coding genes are so conserved that they have very defined functions. They are not up for the surprises. The infection of all those pathogens, even from intrinsic infections. So we have retroviruses inside our DNA that can translate into protein. Our system has to be protected against them also. Protein coding genes, they have very defined function, but what really evolves is what is really recently integrated. What I'm going to show you is that the entire primates, especially hominoids, including chimpanzee, gorilla, and humans, their genome has been modified by multiple waves of retroviral invasion, and that comprises 8% of our DNA. Whenever the new infection or these kind of surprises come, retroviruses are the ones which respond and defend our system against those things. So the reason why embryo is alive is because of these endogenous retroviruses. And actually, I'm also going to show you that they come in all the flavors. They are good, they are bad, they are ugly. So there are some which are very new, which are very selfish. They still have a potential to jump around. They're mobile DNA, right? So they still have a potential to jump around. So they jump from one locus of DNA to other locus, and they cause double-strand break. The new ones, which has recently integrated, that's a feature of HIV, for example. They cause double-strand break, and host, it has a limitation, right? I can still talk in English.
No expression.
That won't buy you time.
So it's a deja vu, and you know what I want to say. Yes, so I'm going to present that these intrinsic resistance, which is coming from the retroelements, which is in our DNA, against the HIV. So there are some people. There are some people in the world. It's very rare fraction of the people. They have this innate capacity to restrict HIV. So if they get infected by HIV, they don't fall sick. Their CD4 T cells do not die. And what I'm going to show you is that how they control spontaneously or naturally, without any drugs, without any treatment, is because of these endogenous retroviruses which is sitting in our DNA. So that's a classical case of fighting fire with fire. I would start with the fact that we share this planet, which is a planet of viruses. We carry viruses in our daily life every day. And there's these are the viruses which are outside of us, where we carry it in our system, but whether they enter in our cell or not, that is a different discussion. And I'm going to show you that there are certain mechanisms or host comfort why these viruses do not enter in our system. Or they might have entered in the past, in the history, but those people are not, they haven't reproduced. It is because I'm interested in the retroviruses which is inside us. 8% of our DNA, 8% of our DNA is derived from sequences, which is from the retroviruses, as you can see here. While the protein coding genes, they only comprise 1.5%. 6 times more sequences in our DNA that comes from the retroviruses. Why? Because, as you know that, for example, HIV, if it integrates in our DNA and if it passes through the germ cells, then it becomes a part of the gametes, male or female gametes, which means that it can go from one generation to another. And if these viruses, they offer some positive selection over the host fitness, they will be part of the population. And that is how these retroviruses, they use their envelope to fuse and enter in the cells, they reverse transcribe, they get integrated into the DNA, and then they exploit the host machinery, the transcription factors and everything from the host to transcribe as their full-length messenger RNA. That gets translated, they make virus particles, they're ready to infect other cells. In theory, those cells should die. But that is the point I'm trying to make here. Why we have so many endogenous retroviruses is because our ancestors were elite for these retroviruses. And there are many, many families. The human genome has been modified by multiple ways of endogenous retroviral infections. Even before 40 million years ago, 32 million years ago, in the different parallelogram of human evolutionary history. So that means that these individuals who survived, at that time, I believe there was no antiretroviral therapy, right? So those individuals who survived, they were elite for these retroviruses. They're like HIV, they're like any other retrovirus, which is highly infectious. And so the point I'm trying to make here is that human genome not only shares this group 1, 2, 3, not only shares the endogenous retroviruses with other primates, but it also has a specific retrovirus which is specific to the lineage of the human. So lots of human-specific sequences are also derived from endogenous retroviruses. How does this? So this is what I was talking when the PowerPoint was off. So, for example, if something which has infected us very recently, at the end of the day, they're transposons, they're selfish elements. They would love to propagate. They would love to be part of our system from one generation to another. So they would jump around. So they would move from one place to another in the genome. They would cause double-strand break. And then they would cause these unwanted mutations that would cause double-strand break. If host cannot repair those double-strand breaks in a given time, then cell cycle arrests. And if host gives up, then either the future is apoptosis or senescence. Both are detrimental. But if these endogenous retroviruses, if they are in our genome for a very long time, if they are not selected out, if those individuals did not die who had this kind of activity of these retroelements, then it would become a function of the cooperation, where the host would take some regulatory activities of these endogenous retroviruses to perform its own cellular function, or let's say the expression of the genes are driven by these cooperative activity of retroviruses. And then if it is very old enough, then they become hosts. They just lose all the property of retroviruses, they become hosts, they code for the protein, without which it's for co-option, because if you make a deletion or if you make a mutation, the host cannot survive. It is that obligatory for the fitness of the host. And so how amazing it is that the co-option of these endogenous retroviruses, they contribute to the placentation, pluripotency, and immune system. And all three are equally important for the survival of any individual. So the reason we are alive, the reason why we are alive here is because of the co-option of these endogenous retroviruses. And I will show you how do they do that. So from past 7 or 8 years, what I have been researching is showing that this regulatory effect. So why these endogenous retroviruses were selected in our genome? Because more frequently they were integrated near near a protein coding gene. And then they were helping transcribing the protein coding gene. And as you see how strong the regulatory potential is, Yamanaka factors, because of which Yamanaka got the Nobel Prize, this cocktail of transcription factors to make iPSCs or the pluripotent cells, they all bind here. And what we did, I can just summarize very quickly. We just made 3 base pair mutations here on the NANOC binding side. And what we found that these pluripotent cells could not remain pluripotent. They either differentiated or they died through the programmed cell death. And one of this intrinsic defense system, how it evolves, is that when embryo gets the embryonic genome activation and the whole genome gets demethylated, then this young elements, which I was talking about, which are selfish elements, they jump around a lot. So those cells, those single cells, which do not have this regulatory activity of HERV, they do not have induction of this ApoBec gene. It drives the expression of ApoBec gene. ApoBec is RNA editing enzyme. It edits the messenger RNA of these bad guys, this retrotransposon, and it doesn't let it transpose and protects the cell from genome instability. So in a way, indirectly, what it does is that it stops the high DNA damage and programmed cell death. And that is why the pluripotent cells are protected because of this endogenous retroviruses. Otherwise, these cells would not exist. And pluripotent cells occur only once in the human embryonic development. And they are defined by the activity of these endogenous retroviruses. Next, the premise of the whole talk is based on this amazing data what we have got. Is that, so what I showed you just now is the impact of this endogenous retrovirus at the DNA level, at the chromatin level. But here, this suppressin locus, we found that this is also endogenous retrovirus. It makes the envelope protein. It makes this viral envelope protein, like how the HIV's envelope protein uses cell receptors to enter in the cell. It makes this, and it is antagonistic of this receptor, which is AST2. So what it does is that if we knock out this suppressing locus, then this exogenous retrovirus infects with more than 80%. And then when we overexpress this suppressant in those cells where this is not expressed, then it becomes resistant to the HIV infection or any kind of retroviral infection. And then we show with the physical binding that it binds to the receptor of this exogenous virus. So what it means, from our DNA, we encode certain protein that binds to the receptor that escapes the binding of retroviruses which is outside of us. That made us think that is it only one instance or there are like more proteins? Because just remember, the protein coding genes, what we know in human genome, how they discover it, there's one criteria that tests whether it is conserved through the phylogenetic tree. And I must remind you that these are relatively very young, so you don't get conservation of them. So we thought of the re-annotating the human genome with protein coding potential, that how many envelope proteins we can have more. What astonished us that, based on DN by DS and all this phylogenetic approach, protein coding potential, and reanalyzing the Max-Planck data, the high throughput Max-Planck data, Silek data, we found that there are like thousands of them. But what was interesting here is that when we looked at the expression of these thousands of unannotated envelope genes in human genome, mostly they were specific to the placenta, primordial germ cells, or pre-implantation embryos. So what happens in somatic cells? Why do they not express somatic cells? Somatic cells also get infected, right? They're also equally prone to the viral infections, as we know that we live on the planet of viruses. Why does it happen there? And then we thought that maybe they will not be activated until you give some metabolic challenge or you give some viral infection. So we tested this idea, and what we did is that, okay, that's why I was using my computer. So let me just describe. So we gave HIV infection and we challenged it by the interleukin-2, which stimulates the CD4 T cells. What we found is that like 53, these envelope genes, they were highly expressed only when the HIV was infected, not when you stimulate the cells with some other mutagens or some other immunogens. And this is, and why it is very important is because when you give antiretroviral therapy, you eradicate the HIV, right? But what HIV already has done to your system, that may be irreversible. So all these envelope proteins, and they crosstalk from one cell to another. It's not really important that it is coded from the CD4 cells. It can go to the B cells and change the transcriptome of B cells. And those individuals can be prone to B cell associated disease. And this is what we tested, and we found very interestingly is that the fact, and the statistically significant fact is that the people living with HIV, doesn't matter they cleared their HIV proteins from the system or not, people living with HIV has a higher risk of lymphoma. And now what we show here, it is unpublished, and this one please don't read. What we show is that after HIV infection, there is an activation of all the GAG and envelope open-reading frames and transcripts, which alters the transcriptome using the receptors in the B cells, and that is kind of irreversible. Even if you give the antiretroviral drug therapy, it is irreversible. The train has left the station. So what could be the real therapy of HIV which does not have long-term side effects? Maybe we can learn from elite controllers. What is the biological mechanism of elite controllers? How do they control HIV without any drugs or without any treatment? So this is what elite controllers HIV infection looks like. Like everybody else, they also get a high viral load. Okay, so they get a high viral load, but they can control this spontaneously. Okay, let me just show you quickly that in elite controllers, what we found, these human endogenous retroviruses that is associated with innate immunity are highly upregulated. And now if you look at the protein coding genes, what is interesting is that antiviral factors are upregulated in elite, and this is coupled by proviral factors, which is downregulated in elite. And one of the proviral factors is CCR5, which is the classical receptor of HIV entry. Let's look at the genome-wide association study. The variant which is most associated with the viral load, which means people who have this SNP, they do not have much of the viral load. What we found is that this is also endogenous retrovirus. The most significant variant of our genome that is inversely proportional to the viral load is basically endogenous retrovirus, and this SNP creates a transcription factor binding site. If the SNP was not there, there was no transcription factor binding site and expression was low. So since this SNP creates the transcription factor binding site, it triggers the expression of this gene. What it does is that it creates the chromatin accessibility, so all these transcription factors bind there, and the expression of the gene of this neighbor gene in elite is way higher than in non-elite. And we have so many different examples like that. The Smitha Badrinathan from Tübingen, she had validated GBP2 and GBP5 that because of this endogenous retrovirus, this gene is induced upon infection and confers the resistance. It does not let enter the HIV inside the cells. So just don't get mad at me, sir. This is last slide, and this is the model I was very curious about. And these kind of mechanisms doesn't let me sleep in the night, and I will be working all my life on that. In elite controllers, the certain mutations just remove the depressing machinery. So in normal individuals, in non-elite people, these HERVs are depressed with the CRAB zinc finger proteins. So this is not expressed, the antiviral genes are not expressed. In elite controller, this machinery is escaped because of that mutation. And then you have trigger of the antiviral genes. And finally, we have intrinsic resistance of HIV, and that is the molecular criteria to define elite controllers. So I'm not going to read that. Just one punchline that every human that is surviving is a success story. Every virus that is alive is a success story. And all the point I want to make is they have done it together. So with this, thank you so very much. And I cannot be all the mentors. And it's really a pride, pleasure, and
thank you very much. Good work. Any observation? Quick questions, please. I have instructions here that questions only for 3 minutes. Okay? Please don't present us. Ask a question. Very crisp.
dead quick question. Do you think this is a mechanism which is conserved across other species who are also infected by viruses?
This is very interesting. It's a good question, ma'am. So, so as I showed, every primate species. So we also looked into rhesus and rhesus macaque, and also. So they have their own defense mechanism, right? So for example, against HIV, what monkeys have, they have this prim 5 alpha that confers the recession, but that is in all the monkeys. So all the rhesus cannot get infected because in humans, prim 5 alpha was mutated. It's a hermit Malik force, right? So it got mutated in human population, and that led the evolution of a virus which caused the biggest pandemic of virus. So every species has their own defense mechanism. This is the human one, which is quite human specific. We don't know about other hominoids like orangutan, chimpanzee, or gorilla, but we know that in rhesus macaque this does not exist.
Yeah, so, hi. So this element, this HERV that you identified, this is HERV-K HML2 element?
No, HERV-K HML2, actually this is very interesting. So HML2 HERV-K is one of the youngest ones.
Yes.
Right? So they have high similarity with the HIV. They have not been co-opted yet for regulating the neighbor genes. They also encode their viral subunits and all. So this is not the HML2 HERV-K. This is something.
Okay, so then what that would indicate is that this particular element is present in other hominins as well, right?
Chimpanzee, gorilla, orangutan.
Right. So if it is so conserved, why don't we see similar responses in non-human primates? Because in those primates, the viral loads are actually very high, no?
For the HIV, you mean?
For HIV-like retroviruses, so for the simian retroviruses, those viruses do multiply, right? So your method here is that because it triggers retroviral genes, those viruses, the viral load would be lower, right?
Yes. Okay. So now,
Thank you.
Yeah, that's a good question. So let's imagine this genomic locus, what I was talking about. Even though this element is conserved across the phylogenetic tree, this genomic locus, where it is in humans, that is not there in rhesus macaque and other species. So what it means is that due to some selection pressure or something, they had been deleted out, but somehow the human system retained them. And I call it addiction model. These endogenous retroviruses, they offer so much in early embryonic development that whenever this metabolic challenge or infectious challenges, the host gets later in the life, they just go back to their embryonic development mechanism to again protect using the same mechanism. The embryonic development of chimpanzee, gorilla, and other hominoids is quite different in this perspective. They have a differential regulation of retrotransposition. Retroviruses act completely different. So as I said, every species has their own defense mechanism. This is quite human specific as far as I know. We have never tested in chimpanzee, gorilla.
Yes. Thank you, Dr. Singh. Excellent work, excellent presentation. Thank you for your cooperation.
Thank you, Dr. Singh, for such an insightful talk on the role of ancient viral infections and the interplay between the HERVs and the modern human genes. I would love to repeat history and request Dr. Rao to present the memento to Dr. Singh, and hope you can use this picture in the future presentations as well. I have a quick announcement to make. We'll have a group photo outside the main entrance.